Hi everyone, my name is Sarah and I'm a postdoc at the University of Wisconsin-Madison in the biochemistry department. Today I want to talk about my lab's work based on a gene called Stearoyl-CoA desaturase 1 or SCD1 and its role in metabolic disorders. This includes everything from diabetes to obesity to fatty liver disease, etc. So from this talk, I hope you learn what SCD1 does in the context of metabolism and how it's currently being studied to better understand metabolic disorders. So generally when people think of metabolism, they think in terms of having a slow or fast metabolism, usually referring to weight loss or weight gain. But from a biological standpoint, metabolism is the sum total of chemical reactions required for an organism to live and involves various cycles of synthesis and breakdown of molecules to meet the needs of the organism. So you can sort of think of your body as if it's a factory with a goal of survival. Metabolism would be all of the daily operations of that factory. To run this factory, you require fuel and building blocks in the form of food. You have some capacity to store energy, and your factory also generates waste. Your tissues, organs, and systems would be interconnected departments that do specific work using machines, and at the smallest level, these machines are enzymes. So enzymes are specialized proteins that help facilitate chemical reactions. For instance, SCD1 encodes a gene, a delta desaturase enzyme that is necessary for converting saturated fatty acids into monounsaturated fatty acids, or MUFAs, by inserting a single double bond into the chemical structure. So metabolic disorders occur when there's a problem in these daily operations as a result of environmental factors, genetics, and mal or overnutrition. So my lab focuses on the overnutrition that results from Western-style diets, particularly ones that are high in carbohydrate, and that specific carbohydrate is sugar. So when we eat carbohydrates, they're broken down into glucose, which can be used by our tissues as energy, or they're stored as glycogen with the aid of insulin and stored in the muscle and liver. Beyond that, excess carbohydrates are converted into fatty acids that are then transported to adipose or fat tissue. In some cases, that fat is also stored in the liver, which results in non-alcoholic fatty liver disease. So how does SCD1 fit into this? SCD1 enzyme is actually activated by carbohydrate consumption. So Western diets are calorically dense and rich in both carbohydrates and fat. With an excess of fatty acids, either made by the body and processed by SCD1 or consumed as dietary fat, these can ultimately lead to metabolic disorders such as obesity and diabetes. SCD1 is actually found throughout the body, including in the liver, adipose tissue or fat tissue, skin, and muscle. So what happens when you get rid of SCD1? And why am I asking this question? Well, a key way to study how an enzyme works is to actually get rid of it. My lab was able to generate was able to study SCD1 deficiency by systematically altering the gene in different tissues in the mice. And we saw some pretty interesting results. So a normal mouse that is fed a Western-style diet that is high in carbohydrate or high in fat will eventually become obese and have insulin resistance, as well as a fatty liver. A mouse that has a whole body SCD1 deficiency, however, meaning that the SCD1 enzyme is not made anywhere in the body, these mice will actually have a normal body weight, have increased energy expenditure, and will also remain insulin sensitive, as well as have no fatty liver when they're fed a high carbohydrate or high fat diet. When we look at a specific tissue, the liver alone, and we are able to have an SCD1 deficiency only in the liver, we also find that these mice are protected from high carbohydrate diets. So they don't become obese or insulin resistant or get fatty liver. The liver is of specific importance because it's a hub for metabolic pathways in the body. So by studying this, we can study sort of the role of SCD1 on whole body metabolism. So you might be thinking, okay, too much SCD1 is bad, and if we just get rid of SCD1, then we've cured diabetes, we've limited obesity, and there's no fatty liver. But there are also issues with SCD1 deficiency. Previous work in the lab and my own has shown that SCD1 deficiency in the liver can also cause early signs of liver cirrhosis, which is scarring of the liver, as well as precursors to hepatocellular carcinoma, which is a type of liver cancer common in people with liver disease. This is because SCD1 is required to make oleate, a MUFA that reduces inflammation and cellular stress that can result in chronic disease. So these results basically show that SCD1 is somewhat of a double-edged sword because too much or too little SCD1 activity can be bad for metabolic health. But this does show that SCD1 is an important regulator of metabolism of fatty acids in the body. So let's go back to our factory. I mentioned that enzymes do work like tiny machines. Sometimes these machines operate like assembly lines, where the output or work of one machine actually influences the input of another. This is sort of analogous to molecular pathways. So in some cases, we don't yet understand all the players in a molecular pathway. So by studying SCD1, we can identify other proteins and other molecules in pathways that may be useful to understanding and treating metabolic disease. So I hope that I've imparted some knowledge about SCD1, and I thank you for listening.